Um, Today I'll be presenting. I am Dr. Sankar, and I'll be presenting on the convective heat transfer on stenosed blood flow through permeable microcirculation in the presence of a magnetic field. So this work was done together with two of my colleagues from the department, Dr. Rao Gunakala and Dr. Kamijong from the Department of Mathematics and Statistics. So did you know that in the Caribbean region, Trinidad and Tobago has the highest overall percentage of deaths due to the chronic non-communicable diseases? The four major of these are the heart disease, diabetes, cancer, and stroke. Under heart disease, examining blood flow through a blood vessel with stenosis is crucial in understanding how blood flows in such patients that have arteriosclerosis, which is a narrowing of the arteries, well, the blood vessels. So in addition, under disease conditions, it is possible for varying the haematocrit level in the blood. For example, diseases such as polycythemia, hypertension, or sickle cell. The haematocrit level in blood is varied. There are also popularly used therapies for heat, in which involve using heat in the medical field to heal injured muscles. And cells and organs are extremely sensitive to temperature changes. Magnetic fields are also in the medical field, for example, in the MRI machines, in cancer tumor treatment and tracers. And there are also certain drugs that can affect vascular permeability. That is the permeability of the vessel walls. And such drugs are like the ACE inhibitors, sildenafil, decongestants, antihistamines. So what this study did was we modeled the situation. So we modeled the stenosed blood flow in the presence of the permeable walls, the external magnetic field, and convective heat transfer. We did this to examine the effect of varying different parameters on the friction factor Reynolds number, temperature, and the Nusselt number. In so doing, this aids in understanding the effects of wall permeability, a magnetic field, and the presence of heat transfer on different disease arterial systems in the future. So here we have the mathematical formulation. The geometry of the flow, we model the vessel as being circular in nature. In the RC polar coordinate system, we consider the vessel of length L, the stenosis beginning at Z is equal to D to Z is equal to D plus L naught, with a corresponding height of the stenosis being delta. And we have the corresponding radius of the different regions. So to model blood, due to observances from experimental data, etc., there's a core region which contains the red blood cells, which we are looking at in the study, the haematocrit. So our model included the core region being a particle fluid suspension and a thin outer region near to the walls. So what we did was a two-phase and two-phase in the center, as well as a two-layered approach to blood flow. So in addition to this, we considered now the permeability of the walls and the resulting applied magnetic and convective heat transfer in the system. So to examine the geometry of the stenosis, what we noticed in different studies or as well, that the stenosis in the upstream is more a straight path, whereas the downstream is more curved in nature. So this is our model that we chose to represent the geometry of the stenosis developed, and this is termed a composite stenosis. So to this, we had our governing equations. Again, as we mentioned, the inner layer of the blood flow was a two-phase particle fluid suspension, whereas the thin outer peripheral layer was a Newtonian fluid. And to this, we have our external magnetic field acting. So we proposed again the corresponding boundary conditions for velocity. So incorporating the convective heat transfer, we also have the suitable temperature equations and boundary conditions. Now, these equations presented here will, after using the simplified assumptions of the system and non-dimensionalizing, which introduced certain dimensionless parameters. So for example, in the governing equations, we got this M, which is referred to as the Hartmann number, and square root Darcy, the Darcy number. Right, so based on our results, we also obtained the mean velocity and the friction factors, Reynolds number, and the Nusselt's number. So these were then calculated and examined. So what we noticed in our results for the friction factor Reynolds number, we increased the hematocrit to C and the Hartmann number M. So what we noticed is that increasing these variables, increasing C and M, resulted in an increase in the friction factor Reynolds number. So just to remind you that C refers to the blood cell count, so our red blood cells, and M is related to the strength of the magnetic field. So as these were increased, our friction factors Reynolds number increased, as well as if we had increase in the stenosis height delta and our square root Darcy, which is related to the permeability of the wall. As these increased, we also noticed an increase in the friction factor Reynolds number. So this friction factor Reynolds number is inversely proportional to the velocity. So what this gave us as well is an idea of what was happening to the velocity of the fluid as we increased these different parameters. So what we noticed again is that if we increase these four parameters, it resulted in a decrease in the velocity. So in certain circumstances, for example, in surgeries, there may be a need to decrease the flow of blood, and the case with increasing the magnetic field slightly, we saw a drop in the velocity. We also examined the temperature. So we increased the hematocrit, the C, the Hartmann number M, and these increased the temperature of the blood, right? So again, those diseases with high hematocrit, or if we keep increasing the intensity of the magnetic field, these would result in an increase in the temperature. And as the wall became permeable, there was also an increase in temperature. Upon examining the Nusselt number decreased with increasing hematocrit and the Hartmann number. So what we noticed as we increased C, they decreased, right? In both scenarios here, the Nusselt number decreased. So what the Nusselt number tells us about is about the loss of heat and energy from the system. So what we noticed as C increased, these decreased. So there was less heat and energy loss. And this was also noted with respect to increasing the stenosis height and the Darcy number. So stenosis height again refers to, you can think of it as, the vessel, the stenosis growing and the pathway through the vessel decreasing. And of course, the Darcy number, we said, was related to the permeability. And we mentioned that certain drugs have the capability of affecting the permeability of the vessel walls. So in these scenarios, what we noticed, Yeah.
1 minute.
Yes, so in these scenarios, we noticed that in patients with elevated red blood cells or those exposed to the high intensity, we had a rise in the temperature, increasing wall permeability, increase the temperature. So this should be monitored in patients who take drugs to affect wall permeability. On introducing the magnetic field and increasing its intensity, there was less heat and energy loss, and the rate of heat transfer decreased. As the wall became more permeable, as hematocrit increased, we had a slight decline in heat and energy as well. So these were the references, and I now welcome any questions related to my presentation. Thank you.
Okay, thank you, Dr. Alana, for the presentation. So any question from the audience?
We have one question here from Ashrad. Well, first one is from Anthony, actually. Did you consider the effects of temperature distribution in living tissue for the analysis?
So in this, we actually just looked at the vessel and not as it relates to the effect on the outside tissue. So not in this study. We just looked at in the vessel itself, microcirculation in particular.
Okay. The second question is from Ashrad Mohammed. He compliments you on your presentation, and he just wants clarification that, or if he had missed something, does the model apply to all blood vessels regardless of the location in the body?
No. So this one in particular is for the microcirculation, which are the smaller vessels, the capillaries, etc. In larger blood vessels like the arteries, they can just be modeled, or it's sufficient to model those as a Newtonian fluid. But in the smaller vessels, different the behavior of blood is different. So this model that we chose is suitable for modeling flow in microcirculation. So just to answer, different parts of the body require different models.
Thank you. And from Sarah Ruplal, she asked, as there was a presence of stenosis in the blood flow, was there any heightened pressure compared to regular blood flow? Oops, just hold on. That may have affected the temperature and by extension, convection during the permeable microcirculation.
Okay, so yeah, just the middle part. Yeah, thanks.
Just the middle part. As I'll repeat the entire thing. As there was a presence of stenosis in the blood flow, was there any heightened pressure compared to regular blood flow that may have affected the temperature and by extension, convection during the permeable microcirculation?
So remember, as the flow is passing through the stenosis as well, there's a narrowing that would occur at that region. So that in itself would affect, just instead just of a regular laminar flow, that narrowing would have an effect on the system. But we didn't really look at, particular what would be the pressure distribution. We more looked at the temperatures and the velocities through the system in this study.
Okay, thank you very much.